Now here you can see the DNA structure as we had done in our earlier classes. They are double stranded. Okay, DNA structure is a double stranded polynucleotide chain. Now this it is a double helical structure. It was proposed by Watson and Crick. Now the chains are anti-parallel polarity. Now if you can focus here, you see one goes from 5 to 3 end and it is 3 end to the 5 end. Okay. Now here you see it forms the backbone of the DNA molecule, DNA chain rather, polynucleotide chain. This is the ribose sugar, a phosphate, and this is the nitrogenous base ATTAGC. Okay, this is a DNA. It is a double helical structure. Now here I'll give you a proper way. You see, adenosine is always joined to thymine with a double covalent bond. Guanine is joining with cytosine in a triple covalent bond, but in case of RNA, the thymine is replaced by uracil. That means AT is in DNA, so if it is in RNA, it is AU. G and C is common to both. Now you see, another thing is very important is first we have to know how does it replicate. I'll give you a short one because in the next part of the chapter, when we have a detail, we'll come to it. See, DNA is basically, it's a double helix structure. This is the carbon sugar backbone, and these are the nitrogenous base that likes the steps of a ladder. Okay, now here you see when the DNA replicates, it's just open like this and part of it copies another daughter DNA like a template. Okay, so this is how DNA replication is there. Now you see, now the next important topic what we will be dealing, rather, it is a packaging of DNA helix. So the packaging of DNA helix is actually the nucleosome model. Now what is this nucleosome model? It is basically a bead and a string model. Now if you see a bead and a string, okay, beads are small structures and string, you know. Now these round structure is basically is of a protein, that is octamer, histone, protein or histone, that is 8 molecules are there, and you see over this protein, that is histone, and over this 2.5 pair of coiling DNA. So together they form the chromosome. Whatever you see, the chromosome or the chromatin material, basically it is an admixture of protein and the DNA coil. Okay, so this is visible under the microscope only when the structure is very condensed and it is in the metaphase stage. Now if you can see, if you open the full string, it is 2.5 meters of chromosome fiber that is coiled and it is found in a just inside the cell. Okay, so it's a very complicated structure, but if you stain it, at certain portions you find it is stained light and some portion it is dark, so that gives you the banded structure of the DNA. The light portion that is stained, it is a non-histone and we call it as euchromatin, and the dark portion that is known as heterochromatin, that takes a dark stain. So if you see, this is the structure of the basic DNA, and here in your this diagram you can see basically it is a method by which it is coiled. Adenine is paired with thymine and guanine is paired with cytosine. These are attached by hydrogen bonding. Adenine with thymine has got two hydrogen bonds, and guanine to cytosine has got 2 bonding. Okay, now another thing is here you can see RNA. That means RNA replication is there, but when RNA forms the mRNA, it is through transcription, and mRNA forms the protein. So this is known as a central dogma of protein synthesis, and it was through Francis Crick. You don't have the detail right here, only this you understand. When DNA is converted to mRNA, it is known as transcription, and when mRNA converts into protein, you call it as translation. Okay, now here you see that how is the DNA helix packed inside the chromosome, inside the nucleus. Okay, now you see one histone and 2.5 circle of the DNA is basically known as a base pair. Okay, so distance between two consecutive base pair, or in a common language between the two beads, it is 0.34 nanometer. So what is 0.34 nanometer? 0.34 into 10 to the power 9 meters is the length of the DNA double helix. Is a typical mammalian cell is calculated simply by number of base pairs. For example, it is assumed that number of base pairs is 6.6 into 10 to the power 9 base pair into the distance, that means 0.34 into 10 to the power 9. So you have a 2.2 meters. So length is a far greater than the dimension of a typical nucleus. So you can imagine how tightly it is packed within the cell. Okay, now here in see, you can see it's like a beaded string or a beaded garland thrown away, but it is basically the beads what you are visible is basically the nucleosome or the histone protein molecule. Now this is made up of 8 octamers, 8 amino acids. Okay, basically the amino acid that is here, it is lysine and arginine. These are the protein. You see, histones are rich in basic amino acid. That means if you stain with the acidic dye, it will turn red. Okay, so that is why it is known as, it stains with a acidic dye, so it is known as basic. So basic amino acids, this residue lysine and arginine, and both the amino acids residue carry positive charge. Now you see, they are positive charge and DNA helix is supposed to be a negative charge. So that is the reason these beads are histones are organized to form a unit of 8 molecule called histone octamer, and the negatively charged DNA. See, DNA is negatively charged and histone is a positively charged. So obviously the chemically neutral when they are attached together. So DNA is wrapped around the positively charged histone octamer to form a structure called nucleosome. Okay, so nucleosome consisting of repeating unit of a structure in the nucleus called chromatin. Okay, now thread-like stain-colored bodies, it is known as a bead on a string model. Theoretically, how many such bead nucleosome you can imagine? So it is a packing of chromatin. So packing of chromatin at higher level requires additional set of protein that collectively referred to as euchromatin and heterochromatin. Okay, now you see, if you stain it, as I said earlier, the non-histone chromosomal NHC protein, it has got a lighter stain that is known as euchromatin, and heterochromatin is a more densely packed stain, dark part. Okay, so euchromatin is said to be transcriptionally active. So the lighter portion, this which stains lightly, is chemically active. Okay, whereas heterochromatin is basically inactive. Now you see, there are certain experiments to prove that what is the genetic material. Okay, so in that, a transforming principle, it was by Griffiths. He practiced a series of experiment. Okay, he performed a series of experiment with what? With pneumonia bacteria. Okay, Streptococcus pneumoniae. Now what he did, he witnessed a miraculous transformation in the bacteria during the course of experiment. What he did, he took 2 strains of bacteria. Okay, one had got a smooth colony, shiny colony, so he named it as S strain, and another one has rough colony, so it is known as R, because they are clusters, bacteria cluster. So he took 2 strains, S and R. Now this is because S strain bacteria have a mucus polysaccharide coat, so that gives the shiny one. So R does not have the coat, so he infected the mice with S strain. S strain was, and it was virulent. Virulent means infectious. So it resulted in the death from the pneumonia infection. But mice infected with R strain did not develop pneumonia. So it was a proven fact. Now what he did, see, you can see it on the screen. S strain, when injected with mice diet, R strain injected live. Now what happened? He boiled them. So when he boiled, S strain was killed. So when injected, mice was alive. But when S strain killed plus R strain was alive and it was injected, the mice died. Why? Because heat killed some of the protein structure. Okay, but you see the R strain got transformed. Okay, so R strain got transformed by the heat killed S strain. So such transforming principle transferred with the heat killed strain enable the R strain to synthesize. So smooth polysaccharide, so it got, so biochemical